tools and technology in. Next topic is chromatography techniques. Chromatography techniques. Chromatography is a biochemical technique introduced by Michael Pitt in 1906. Originally, this technique is used to separate the different color pigments of mixture. Chromatography to write. This technique has undergone various modifications, and now this technique can be used to separate all compounds, whether color or colorless. Chromatography is defined as the technique of separation of substances according to their solubilities in two immiscible phases. Already we know two miscible liquids are there, immiscible liquids are there. Here we are using the immiscible liquids. Okay, so immiscible liquids in, we can separate the two substances due to the chromatography techniques. This procedure of separation is called chromatography. This procedure is known as chromatography, and the preparation is called chromatogram. This chromatography preparation is known as chromatogram. Chromatography is a separation technique is used in chemical and biology, in chemical also and as well as biology also. Okay, so next, chromatography is the separation technique is used in chemical and biology. This technique is used for separations and identification of mixtures of chemical compounds and biological compounds. We can separate the biological and as well as chemical compounds also. Biological compounds means different types of pigments are there. We can separate that. Now, for example, pigment means what? Color pigments are there. So that color pigments also no problem, or otherwise we are using in chemistry azo compounds also no problem. So that type of two compounds we can separate. Means the in chemical in a sample, some laboratories are giving to two mixtures in a one sample. So we can separate due to this technique. Okay. So that's why we are separating the chemical compounds and as well as biological molecules also. Components of chromatography include the following. Sample to be separated. Means sample to be separated. Next stationary phase. Next moving phase. Means here samples to be separated. Okay, sample to be separated means here stationary phase is there and as well as moving phase is there. What is the stationary phase? Stationary phase means for example you can take any type of we are taking here silica gel. Silica gel is the stationary phase, means it is not move, means it is not going to move, but moving phase is there. For example, we can take any type of alcoholic solutions also no problem, or otherwise any type of other water also no problem. That is known as moving phase. Means already you know in chemistry previous semester we have to learn about this phases. Means moving phase is there and as well as stationary phase is there. For example, ethyl alcohol we are taking, acetone we are taking. This is moving on the silica gel or otherwise chromatography paper. Okay, so due to this moving phase, so different types of compounds are separated and as well as they are moving from one point to another point. Means previous semester we have to prepare the silica gel slide. Okay, so on that silica gel slide we are uh, taken the different types of proteins. Okay, different types of proteins we are taking. So due to that take proteins, we have separate the different types of proteins. What is that proteins? Alpha, beta, and as well as glycerin and The biological proteins means, for example, cysteine, histamine. So like that proteins were separated. In chromatography, separation of molecules is based on the following principles. Means here the chromatography technique using for the separation, especially separation for the molecules. First one is separation of differential solubilities of sample in stationary phase and in moving phase. Means here we can take the separate stationary phase and as well as moving phase. Means the stationary phase sample you can take and as well as the moving phase sample you can take. And uh, due to the chromatographic technique, we can separate that two. All the chromatography techniques fall into two main categories based on the nature of the stationary phase. The stationary phase, according to stationary phase, chromatography techniques are two types. First one is the adsorption chromatography. Here what? Adsorption chromatography, not absorption, adsorption chromatography. Okay. Second one is partition chromatography. What is that? Second one is partition chromatography. Adsorption chromatography means what? Adhishoshanam. In Telugu, we are saying adhishoshanam. Means the adsorption is takes place. Shoshanam. Okay. So means shoshanam means what? Adsorbed. Absorbed. Okay. Partition. Second one is partition chromatography. Okay. Have understood. Now we can go to adsorption chromatography technique. In this type of chromatography, the stationary phase is a solid such as alumina or silica gel, and the mobile phase may be liquid or gas. Means here we are taking alumina, means aluminum hydroxide we are taking, and that aluminum hydroxide is dissolve into the water, and we can dip that slide here down given. We can discuss about that. So dip into that what solvent. So the aluminum is stick on the slide and we can dry up, then afterwards we can use that slide. Okay. Next, silica gel. Then so silica gel means what? Silicon oxide. Silicon oxide. SiO2. Silicon oxide is dissolved into the water, so it is known as silica gel. Okay. And the mobile phase. Means this is the one of the. In this one is the mobile phase, and so another is the stationary phase. Means the alumina and as well as silica gel is the stationary phase. Next, mobile phase is known as liquid or gases. We can take liquid also no problem, air gases also no problem. Liquid means here especially acetone is taking, and as well as ethyl alcohol is taking. Next, sometimes we are taking the different types of alcoholic substances also no problem. Means butanol also no problem, or pentanol also no problem, ethanoic acid also no problem. Like that substances we are taking. Next, gases phases means for example the ammonium gas also no problem, and as well as HCl gas also no problem. Like that gases we are taking. Next, second one is partition chromatography. In this chromatography, the stationary phase is liquid and mobile phase may be liquid or gas. Means partition chromatography in stationary phase is a liquid and mobile phase is liquid also no problem or gases also no problem. Okay. Next, the paper chromatography. This is shortcut we are saying PC. Okay. Paper chromatography. This technique was introduced by Schieber and Imzilo. What is that? Schieber and Imzilo in 1938. It is a type of partition chromatography. This is known as partition chromatography. In this technique, a strip of paper is used to support the stationary and water phase. Hence the name is known as PC, means the paper chromatography. Means here only we are taking tissue paper. Okay, which type of paper? Tissue paper we are taking, and were dipped into the different types of substances or otherwise solvents also no problem. In this technique, a strip of paper is used to support the stationary water phase, hence the name is PC. The mobile phase is also a liquid. Mobile phase also a liquid. Separation is based on liquid-liquid partition of the compound. So here black diagram is given. This is the spot of ink, means the ink we can taken here spot were taken, line drawn by pencil. This is the line, means you can take the 1 centimeter or otherwise 5 centimeters. You can mark it because of how much substance is spread on this slide. Okay, strip of filter paper, strip of filter paper. Okay, so here we are taken a empty beaker or otherwise you can take a jar and you have to take water. Okay, water is the stationary phase. Next spot is kept already here shown, that is the spot. So ink spot were taken, then afterwards you can dip into this water and hang with the glass rod. Okay, this paper is hang with the glass rod. Paper clip is kept because of it is not fall into the water. Next, this is known as a beaker or otherwise you can take jar also no problem, measuring jar. In your laboratory what is there, you can use that strip of filter paper. Okay, the following the important type of PC, ascending PC. Next, second one is descending PC. Next one is radial PC. PC means paper chromatography. Ascending paper chromatography. In this technique, following the steps are involved. First one is take Wattman number 1 filter paper. Means in our laboratory, Wattman number 1 filter paper is there. You can take that filter paper and you can cut into the size of 2 into 10 centimeters. Okay, and draw a line about 3 centimeters from one end. From one end you can take 3 centimeters because of the substances how much spread onto the filter paper. Next, place a very small spot of mixture to be separated at the center of the line. Solvent system moving phase. Solvent system moving phase is prepared and taken in the chromatography chamber. Suspend the paper strip in a through in such a way that the paper edge is just below the solvent level and the pencil line should be above the solvent. Means we can dip into this spot into the solvent, just edge of the paper. Okay, the paper should not touch the sides and it should not be slatted, means that paper is not touched to the jar of the sides. Okay, jar of the sides, and as well as it should not be slatted. Okay, keep this setup undisturbed for some times till the solvent rises and reaches the top. Means this this setup you can keep into the undisturbed for some times. Then afterwards you can use the formula and calculate the RF value. Okay, RF is equal to distance traveled by the sample by distance traveled by the solvent. Okay, each spot can be identify the comparing the RF value with the standard RF value. Okay, most of the compounds are colorless and location reagent is applied by the spraying on the paper to detect the spot. Means, then spread after you can take the location reagents. reagents means, for example, we are taking different types of reagents, because of we can separate that compounds. For example, I will say, you can take an primary amine, if you can think primary amine. So here we can take Hinsberg reagent. What is that? Hinsberg reagent. In the third sem, we have to learn about the Hinsberg reagent. Hinsberg reagent is identify the different types of amino acids. So that type of reagent you can take and spray on the paper, because of the color will be changed. So due to that color will be changed, we can identify easily the which amino acid is spread on the paper. So easily we can find it. Okay, next, second one is thin layer chromatography. This is the recent modification of the column and paper chromatography. This is the recent modification. This is the modified chromatography technique. It is a new technique. Okay, so column and paper chromatography. Now up to now we are learned about the column chromatography and paper chromatography. That chromatography too, this is the modified chromatography. In this technique, a thin uniform film of absorbent silica or alumina containing a binder calcium sulfate is spread onto a plate. The thin layer is followed to dry at room temperature and is then activated by heating in an oven between 100 to 250 temperature. In an oven, we can dry up that filter chromatography. Thin layer chromatography about given here. See, the activated plate is 10 activated plate is then placed flat and samples are spotted with the micropipette carefully on the surface of thin layer. ఇక్కడ ఏం చేస్తున్నామంటే ద యాక్టివేటెడ్ ప్లేట్ ప్రిపేర్ అయిన ప్లేట్ ఏం తీసుకొని ఏం చేస్తున్నామా, ఫ్లాట్ గా పెట్టేసేయాలి. ఫ్లాట్ గా పెట్టేసి దానికి ఏం చేయాలంటే స్పాట్ ను అరేంజ్ చేసుకోవాలి. నెక్స్ట్ మైక్రో పిపెట్ ను తీసుకొని, మన ల్యాబొరేటరీలో మైక్రో పిపెట్ ఉంటుంది, ఆ మైక్రో పిపెట్ ను తీసుకొని కేర్ఫుల్ గా దాన్ని ఏం చేయాలి, మనం సర్ఫేస్ ఆఫ్ ది థిన్ లేయర్, సర్ఫేస్ ఉంటుంది కదా, దాని సర్ఫేస్ దగ్గర మనం స్పాట్ ను పెట్టేసేయాలి. దెన్ ఆఫ్టర్ ద శాంపుల్ ప్లేస్మెంట్ చేసుకున్న తర్వాత ఏం చేయాలమ్మా, ద ప్లేట్ ఈస్ ప్లేస్డ్ వర్టికల్లీ ఇన్ ఏ గ్లాస్ ట్యాంక్ కంటైనింగ్ ఏ సూటబుల్ సాల్వెంట్. దాన్ని ఏం చేసేయాలి, వర్టికల్లీ అంటే నిలువుగా పెట్టేయాలి. నిలువుగా పెట్టేసి ఏం చేయాలి, సూటబుల్ సాల్వెంట్ లో పెట్టేస్తే, లేకుంటే సూటబుల్ సాల్వెంట్ తీసుకున్న తర్వాత దాన్ని వర్టికల్ గా నిలువుగా పెట్టేసి మనం కొంచెం సేపు అయిన తర్వాత చూసుకోవాలి. ఓకే, విత్ ఇన్ ద షార్ట్ టైమ్ వేరియస్ కాంపోనెంట్స్ గెట్ సెపరేటెడ్ బై ద సాల్వెంట్ రైజింగ్ త్రూ ద థిన్ లేయర్. కొంతసేపు అయిన తర్వాత ఏమవుతుందమ్మా, అది పైకి ఎగబాకుతుంది. పైకి ఎగబాకిన తర్వాత ఏం చేయాలి, దాన్ని తీసేసి మనం మళ్ళీ డిఫరెంట్ టైప్స్ ఆఫ్ రియేజెంట్స్ ను స్ప్రే చేసుకొని మా కలర్ఫుల్ అంటే ఏ కాంపోనెంట్ ఎంత వరకు ఎగబాకిందో మనం ఫైండ్ అవుట్ చేసుకోవాలి. ఓకే, హ్యావ్ అండర్స్టూడ్. విత్ ఇన్ ఏ షార్ట్ టైమ్ వేరియస్ కాంపోనెంట్స్ గెట్ సెపరేటెడ్ బై ద సాల్వెంట్ రైజింగ్ త్రూ ద థిన్ లేయర్. ఈ టెక్నిక్ వల్ల ఏం చేస్తున్నాం, మనం ఈజీగా మిక్స్చర్ ఆఫ్ కాంపౌండ్స్ ను సపరేట్ చేస్తున్నాం. ఓకే, అంటే ఇక్కడ వెరీ వెరీ సెన్సిటివ్ కాంపౌండ్స్ ఆర్ దేర్ హియర్. ఎందుకంటే మనం ఇప్పుడు సాలిడ్ కాంపౌండ్స్ ని ఈజీగా సపరేట్ చేయొచ్చు. మనం ప్రీవియస్ క్లాసెస్ చిన్నప్పుడు నేర్చుకున్నాం. ఓకే, పికింగ్ మెథడ్ అని. ఇప్పుడు మనం ఏం చేస్తున్నాం, సపరేట్ చేయాలంటే పికింగ్ మెథడ్ ను ఉపయోగిస్తున్నాం, లేకుంటే విన్నోయింగ్ అని చూస్తున్నాం. అంటే చెదగడం, నెక్స్ట్ బండలు ఉంటే తీసి పడేయడం, అట్లాంటి పికపింగ్ మెథడ్ ను చూసినాం. ఇదేంటిదంటే మనకు సపరేటింగ్ టెక్నిక్. ఈ టెక్నిక్ ఎందుకు చూస్ చేస్తున్నాం అంటే మనం ఫర్ ఎగ్జాంపుల్ మెడికల్ ఫీల్డ్ లో పోయినట్లయితే మెడికల్ ఫీల్డ్ లో చూసినట్లయితే మనం ఏం చేస్తున్నాం, మన బ్లడ్ ఏదైతే ఉన్నదో డిఫరెంట్ టైప్స్ ఆఫ్ కాంపోనెంట్స్ తోని ప్రిపేర్ అయింది. అది ఏంటిదమ్మా, అందులో ఆర్బీసీ ఉన్నది, డబ్ల్యూబీసీ ఉన్నది, ప్లేట్లెట్స్ ఉన్నాయి, సీరం ఉన్నది, క్రియాటినైన్ ఉన్నాయి, అన్నీ ఉన్నాయి. కాబట్టి ఒక్కొక్క దానికి సపరేట్ చేసుకొని దాని యొక్క పర్సంటేజ్ ఎంత ఉన్నది అనేది మనం ఫైండ్ అవుట్ చేయడం జరుగుతుంది. ఫర్ ఎగ్జాంపుల్ ఒక్కొక్కలకు కిడ్నీస్ ఫెయిల్యూర్ అవుతాయి. అప్పుడు ఏం చేస్తున్నాం, క్రియాటినిన్ చూడడం జరుగుతుంది. అంటే ఆ తీసుకున్న బ్లడ్ లో క్రియాటినిన్ ఎంత ఉన్నది అంటే ఆ బ్లడ్ నే మనం ఆ క్రియాటినిన్ మనం ఫైండ్ అవుట్ చేస్తున్నాం ఆ ఎంత ఉన్నది అని. ఈ టెక్నిక్ చాలా యూస్ఫుల్ అయినది. అంటే అన్ని లేకపోతే అన్నిటి యొక్క పర్సంటేజ్ చేయాల్సి వస్తుంది. అంటే ఓ 1/1 అంటే ఆర్బీసీ పర్సంటేజ్ ఎంత ఉన్నది, నెక్స్ట్ డబ్ల్యూబీసీ పర్సంటేజ్ ఆ విధంగా చేయాల్సి వస్తుంది. కాకపోతే ఈ టెక్నిక్ వల్ల ఏమైంది, మనకు ఈజీగా మనకి ఏది కావాలనో అదే సపరేషన్ చేసుకొని దాని యొక్క పర్సంటేజ్ను ఫైండ్ అవుట్ చేయడం జరుగుతుంది. దానివల్ల టైం సేఫ్ అవుతుంది, యాజ్ వెల్ యాజ్ మనకు అక్యురేట్ వాల్యూస్ రావడం జరుగుతుంది. ద గ్లాస్ ప్లేట్ ఈస్ taken out from the tank, allowed to dry, and spot are detected by spraying the plate with variety of reagents. Okay, the superiority of TLC, thin layer chromatography, over PC lies in the following facts. Means the superiority, దేనికి superiority ఉన్నది అంటే TLC కా లేకుంటే పేపర్ క్రోమాటోగ్రఫీ కా అంటే ఇక్కడ మనం ఏం చెప్తున్నాం, టిఎల్సి అనే సుపీరియర్ ఉన్నదని చెప్పడం జరుగుతుంది. ఇంకా అంటే పీసీ పేపర్ క్రోమాటోగ్రఫీ తీసుకుంటే రిక్వైర్ లాంగ్ డ్యూరేషన్ ఫర్ సపరేషన్, వేర్ యాజ్ టిఎల్సి రిక్వైర్డ్ షార్ట్ డ్యూరేషన్ ఫర్ సపరేషన్. పీసీని తీసుకున్నట్లయితే లాంగ్ డ్యూరేషన్ అంటే డ్యూరేషన్ ఆఫ్ టైం అనేది ఎక్కువ తీసుకుంటుంది. అదే టిఎల్సి అయితే విత్ ఇన్ మినిట్స్ లో మనకు separate అవుతుంది. ఓకే, separate out the mixture by the TLC, but PC is taken more time, so that's a duration of time is taking more for the PC, duration of time taking is less in TLC is extremely sensitive, which require only small amount of samples. నేను పీసీని తీసుకున్నట్లయితే ఎక్కువ రిక్వైర్డ్ అవుతుంది, శాంపుల్ అనేది ఎక్కువ రిక్వైర్మెంట్ అవుతుంది. కాకపోతే టీసీ అనేది ఎక్స్ట్రీమ్లీ సెన్సిటివ్, ఇది సెన్సిటివ్ కూడా, మరియు స్మాల్ అమౌంట్ ఆఫ్ శాంపుల్స్ మనకు యూజ్ అవుతున్నాయి, అంటే ఎక్కువ వేస్టేజ్ కాదు. టిఎల్సి ఎఫిషియన్సీ ఈజ్ మోర్ దాన్ పీసీ. టిఎల్సి ఈజ్ efficiency is more than PC, paper chromatography. Here black diagram is given, once you can check out. Okay, next we can go to column chromatography. Column chromatography is the technique in this. All the major types of chromatography are routinely carried out using the columns. Okay, using the columns. All the major types of chromatography routine carried out using the columns. Long columns 20 into long columns 20 into 1 centimeter give good resolution of components. Wider columns are preferred for large quantities of samples. An insoluble material is packed on the column and the molecules to be separated are added to the top of the column. The migration of the molecules is started by adding a solvent. The separation depends on the choice of the solvent and carried material. This technique is important for the separation of enzymes such as cytochrome. Means this technique is especially using for the enzymes. Means enzymes separation for this technique is using. Next, fourth one is ion exchange chromatography. Ion exchange is a process in which ions of insoluble substances exchange ions in a surrounding solution. The insoluble substances are called as ion exchangers. Insoluble solvents are known as ions exchangers. Okay, proteins, resins, cellulose, bone, living cells, and silicate minerals and are good example for ion exchangers. We are using proteins also no problem, resins also no problem for the ion exchangers. For ion exchange is process in which ions of insoluble substances exchange ions in a surrounding solution. The soluble substances are called as ion exchangers. Okay. In ion exchange acts as either cationic or anionic exchanges. In this two there cationic exchanges and anionic exchanges. The biological molecules exist in electrically charged forms under different conditions, and these are made use of in their separation by ion exchange chromatography. Amino acids may have negative or positive exchange and can be separated by using ion exchange resins in ion exchange chromatography. Okay. Means here we are taking amino acids, especially positive amino acids and as well as negative amino acids. Okay. Uh, Here black diagram is given, column chromatography for. Okay. See here, ion exchanges chromatography. Amino acids maybe have negative and positive exchange of ions. Here, see black diagrams is given. So these are uh, dots are appearing. That is known as ions. Next, remaining solvent is there. So affinity chromatography. This is the fifth technique for affinity chromatography. Okay, this is a type of adsorption chromatography. There is a high degree of specificity in the interaction between the adsorption and the compound to be separated. In this technique for separation of antibodies, we have to use antigens and vice versa. And see this overall, I will say affinity chromatography is a technique for separation the antibodies. Means in our body how many antibodies are there, how many antigens are there. Means especially this is using for the separating the or counting the antibodies and antigens. For the separation of enzymes we have to use substrates and vice versa also, no problem. This is the highly specific and rapid separation is achieved. Gel permeation chromatography. This is the one more technique for separation the different types of molecules. Molecules are also separated by the basis of differences in their size and weight by passing them down into a column container swollen particles of gel. Means here the the weightful substances will be goes into the down and weightless substances are comes up. So due to that technique we can separate that molecules. Next small molecules can enter the gel but larger molecules are excluded from the cross-linked network. And so due to this, we can separate that molecules. Next here, black diagram is given. Various gels are available for this purposes. Means Here see the molecules of gels and molecules of substance. Next, gas chromatography. Gas chromatography is a separation technique involving passage of gas through a column containing fixed phase. Gas chromatography, liquid chromatography. Okay. Gas solid chromatography, GSC. Gas chromatography means gas solid chromatography. In GSC, the stationary phase is solid and the mobile phase is gas. Already you know, due to the technique, we can separate that gas and solids. Next, gas liquid chromatography. Gas liquid means in this liquid is the stationary phase and gas is the mobile phase. So due to that, we can separate that components. Next, electrophoresis. Electrophoresis is the migration of charged particles molecules in a medium under the influence of an applied electric field. Means here we are using the electrical field, means we can pass the electricity into this. So due to that electricity, electrolysis process is done. So due to the electrolysis process, electrophoresis is based upon the size, shape, and molecule weight of the molecules. So due to that, we can separate that molecules and as well as substances. So due to that, one formula is given. F is equal to delta E by D into Q. Means what is that F? F is the field strength applied. Okay. Delta E by D is equal to field F. F is equal to delta E by D. Q is the charge of the molecule. Okay, I understood. So therefore, due to that, we are getting this formula. F is equal to 6 pi RV, because of R is the radius of the column, V is the volume of the substance, pi is the that means the radius of the that column. Next, 6 is taking for the due to that mobile phase and as well as a film. V is the viscosity, R is the radius of the molecule, V is the velocity of the molecules. Velocity of also given because of this is the centrifugal separations maybe. So due to that centrifugal, we are getting the force. Delta E by D Q is equal to 6 pi VRV. Okay. Next, electrophoresis. Electrophoresis is the migration of the field. Okay, I have understood. Next, factors affecting the electrophoretic mobility: sample, electric field, buffer, medium. What is the buffer? Buffer is the solution we can prepare in our laboratory. Okay, sample. The sample we can prepare in our laboratory. So due to that shape and as well as size, we can separate the electric field. Electric field we can use into the potential gradient, increase the rate of migration. And so the molecules are separated due to this electrical field. Here when 100 MV of electric field is applying, okay, migrate faster than 50 MV, means 100 we are using. Buffer helps in maintaining constant pH. pH is maintaining, here buffer is using. What is the buffer? EDTA also no problem, or citrate also no problem. Citric acid, citrate, formate also no problem. Generally 0.05 to 0.1 m ionic strength of buffer is used. Medium, the supporting medium can exert absorption and molecule effects on the absorption. So due to that technique, we are absorbed by the different types of molecules. So here the absorbance is taking place sharp band, but it contains a tail. Okay, molecular sieving. So molecular sieving supporting media such as polyacrylamide agar. Agar we are using. Already you know what is the agar. Agar is the one of the chemical we can getting from uh, algae. Algae. Okay, in your botany, algae are there. Due to that algae, we are collecting this agar substance. Types of electrophoresis. Electrophoresis is of two types. Free electrophoresis. Second one is zone electrophoresis. What is the free electrophoresis? It is a type of electrophoresis without stabilizing media. Next, zone electrophoresis. It is a type of electrophoresis with a stabilizing media. Free electrophoresis. Free electrophoresis has two main techniques: micro electrophoresis and moving boundaries electrophoresis. Micro electrophoresis. Micro electrophoresis is the technique observe motion of the small particles. Means motion. అవి ఎంతవరకు మూవ్ అవుతున్నాయి దాని యొక్క మోషన్ బట్టి దాన్ని మనం సపరేషన్ చేయొచ్చు. అబ్జర్ప్షన్ స్పెక్ట్రోస్కోపీ and molecular sieving spectroscopy. Okay. Next, we can go to double thing. Here, black diagram C types of electrophoresis for boundaries. Means here the wires are given. It is the anode. It is known as cathode. Since the anode is having positive charge, cathode is having negative charge. Next, buffer solution is using. This is the, if you can think as a cell, this is known as cell. Positive ions and as well as negative ions are there. So when you are passing the electricity, so that cations and anions are getting on the cations are getting on cathode, anions are getting on anode. Okay, minus anode plus cathode. Okay, నెగటివ్ గా ఉన్న ఆయాన్లు పాజిటివ్ సైడ్ వెళ్తాయి, పాజిటివ్ గా ఉన్న ఆయాన్లు నెగటివ్ సైడ్ వెళ్తాయి. ఓకే, హావ్ అండర్స్టూడ్ ఆల్రెడీ యు నో. ఓకే, zone electrophoresis. Zone electrophoresis is of two types: paper electrophoresis, gel electrophoresis. Paper means the apparatus equipments required for zone electrophoresis consist of basically of two types: power pack electrophoretic cells. Power pack means it provides a stabilized direct current control both voltage and current output. Okay, current and voltage are maintaining. Power packs of 0 to 500 volts and 0 to 150 ampere also no problem are available. Electrophoretic cells. Here electrophoretic cells are paper electrophoresis as stabilizing media is filter paper. Here stabilizing media is a filter paper. Due to that filter paper, we can separate that to different types of anions and cations. Paper electrophoresis. In this type of paper electrophoresis, stabilizing media is there. Next, between two buffers reservoirs is there. Next, sample application is important, or otherwise narrow uniform streaks is getting. Okay. Gel electrophoresis based on a type of gel used. It is two types. One is SDS PAGE, sodium dodecyl sulfate and polyacryl amide gel electrophoresis are using. Application: electrophoresis is used to analysis of PCR products. PCR products. Example in molecular genetic diagnostic and genetic fingerprints. Here, this technique is using for the genetic diagnostic, DNA test for, or otherwise any type of other genetic diagnostics for. Means which type of DNA is there, like that. Genetic fingerprints also. Fingerprints, now we are using that fingerprints also. It is used the separation of DNA fragments for DNA fingerprinting. Okay, investigation crime science. It is using in the crimes. Okay. Next, it is used to analyze genes associated with a particular illness. What is that illness? Illness for what is the reason? We can find it. Means sugar patients are there. Next, cancer patients are there. Why the which type of organ for